Welcome to our last keynote speech. It's my great pleasure to introduce Dr. Elaine Mardis. So Dr. Elaine Mardis is currently the Steve and Cindy Rasmussen Nationwide Foundation Endowed Chair at the Nationwide Children's Hospital in Columbus, Ohio. And she's also the professor of pediatrics and neurosurgery at Ohio State College of Medicine. And she joined the Nationwide Children's Hospital in 2016. And before that, she spent over 20 years in WashU St. Louis School of Medicine, where she was Robert E. and Louis F. Dunn Distinguished Professor. And in the past year, she was the president of AACR, the American Association for Cancer Research. And last year, because of her distinguished contribution to cancer genomics, she was elected to the National Academy of Medicine. So when Dr. Mardis was at WashU Genome Center, she pioneered many important work in genomic research and genomic medicine. And in particular, she, I think everyone knows, she co-led a team that reported the first next-generation based sequencing of a whole cancer genome. And over the past decade, she has been playing key roles in large-scale cancer genome projects, including the TCGA. And there's no doubt that she has been a leader in defining and also redefining our understanding of the cancer genome. And her current research interests to focus on genomic and also immunogenomic characterization of cancer and its clinical application to pediatric cancer medicine. And I'm sure she's going to tell us a lot about her ongoing work today. So Elaine, it's really our great pleasure and honor to have you as our last keynote speaker. And thank you for joining this conference, and we look forward to the talk.
Thanks so much, Jan, and thanks for the very nice introduction. And certainly thank you for the opportunity to present this keynote lecture for your attendees at the RSG Dream conference. So I will indeed be talking about pediatric precision medicine today, and hopefully you can see my slides and hear me well. So the title of my lecture today is Pediatric CNS Cancers. So I'll be talking about brain cancers. And I'm really trying to talk about exploring the clinical data, exploring clinical data to inform the treatment of these patients. And what I'd like to emphasize before I get started is just an overarching theme for the lecture, which is really how we can utilize a translational mechanism, namely a cancer protocol that's aimed at characterizing individual patients' tumors for the purpose of identifying perhaps potential new treatments or targeted therapies, clinical trials that they might enroll on, et cetera. In a mechanism where we essentially use the information that's being gained not only to benefit the patient, but also to form hypotheses that may further advance the treatment of patients in the future. And again, with an emphasis on pediatric brain cancers. So why this point of emphasis? Well, in terms of pediatric cancers, as you can see here, brain cancers are now the predominant cause of death by disease in children. So even though leukemias are much more common in their occurrence, brain cancers really have not experienced the advances in treatment and successes in treatment that leukemias have. And this is really now one of our key problems, not only in the pediatric setting, but also in the adult setting. So when we think about the ways in which cancers are classified in terms of pathology-based classification, we typically turn to the WHO guidelines that are put together by eminent pathologists around the world on a periodic basis. These are updated. And CNS tumors are no different. So they are classified largely by pathologic features and cell type of origin. And these are just listing some of the common categories that are identified through the pathologist microscope. But one of the ways that we have been studying these pediatric cancers has really led us to, and others, to new realizations about brain cancers. And in particular, this has been a bit revolutionary in terms of the way that we think about the brain as an immune privileged organ in particular, and how that paradigm may be falling by the wayside. So in particular, what is the cancer immunity connection in the brain? In particular, we know that the immune system can mount a response that leads to killing of cancer cells, and this occurs through a series of stepwise events in a normal setting that are listed out here. We know that disruption of any of these steps of the cancer immunity cycle can ultimately result in tumor growth. And we know that, in fact, there are mechanisms by which these immune responses are suppressed, so-called immunosuppression. And these are typically due to factors that are present in the tumor microenvironment, namely the cell component that is resident within the tumor cells and amongst them, because no tumors are purely 100% tumor cells. And in the setting of the central nervous system in particular, it was long thought that this was an immune-privileged setting, that antigen-presenting cells, T cells, other types, dendritic cells, did not present behind the blood-brain barrier. And we're now realizing, especially since landmark papers like the one cited here from 2015 were published, that there are actually mechanisms by which there is a classic lymphatic system that's located within the CNS. And this might be a mechanism by which the immune-privileged environment of the brain is really not what we thought it was to begin with. And in particular, one of the ways that we're trying to evaluate this is using real data from patients that are being identified with brain cancers, having surgery to remove those brain cancers for curative intent, and providing us with consent to study those using a variety of genomic approaches, which I'll describe for you today. We call this study multiplex characterization of the immune microenvironment of pediatric brain cancers. And one of the reasons for focusing on the immune microenvironment is because, as I'll demonstrate today, and you may already be aware, there are certainly a lot of ways now where immunogenomics, the application of what we know about immune cells to genomic data, can really tell us a lot about these the immune microenvironment in this setting. And also the reason that we're interested in this is because, as all of you are probably aware, immunotherapy as a general broad class of therapeutics is really considered one of the most promising therapeutic classes in cancer medicine today. And I've just listed some examples here. I'll circle back to these types of approaches at the end of the lecture. So this study is really based, as I mentioned earlier, on data from comprehensive DNA and RNA profiling of pediatric CNS cancers. And the idea is to characterize these cancers in a variety of ways, which I'll describe, but really to focus in on characterizing changes in immune cell infiltrates with a focus on what we call longitudinal samples. So namely, a primary tumor and a recurrent tumor that happens at some time interval after the primary tumor is detected, partially or wholly removed by surgery, and there's a subsequent treatment interval. And we really are understanding now more and more about the changes in the immune microenvironment, as I'll try to illustrate for you, in that interval between the primary and a recurrent cancer. So how are we doing this? Well, we launched a protocol with an IRB-approved approach in early 2017 at the Institute for Genomic Medicine. It's a partnership between the Institute for Genomic Medicine and also many of the cancer providers in our hospital across different disciplines, but of course today just focusing on the neuro-oncology space. The eligibility for this study is patients in our hospital with rare, relapsed, or treatment refractory cancers. And the ways that we study these are sort of described in brief bullet points below. So first of all, there's the tumor to normal comparison. This occurs using what I call exome plus. So it's the normal hybrid capture of coding exons across the genome, but includes also cancerous copy number probes that span across all chromosomes at about 30 kb resolution. Using this combination of exon probes as well as copy number probes, we're able to essentially study the majority of the genome without adding a lot of space on top of the normal exome, which is about 1.5% of the human genome. And we can do this at high coverage depth. So this is sort of like whole genome sequencing, but really a much less expensive option and allows us to generate significant coverage depth for looking at heterogeneity, for example. In addition to the DNA comparison of tumor normal, we also study RNA through RNA-seq approaches using multiple analytic pipelines, which I'll describe. More recently, we've added in the EPIC methylation arrays using classifiers such as the DKFZ CNS classifier to type and in some cases subtype the different cancers that we're studying. And then finally, we have a fusion driver identification clinical assay, which is based on the Archer DX platform. One of the things that we did initially and has been really valuable is that we, instead of having on-premises computing, really ported all of our computational pipelines to the Amazon Web Services cloud. And these run directly from our Illumina sequencers using the whole exome sequencing or RNA-seq, as I mentioned already, directly up to the AWS Docker containers that then perform the Churchill 3.0 rapid alignment to the genome and spin out a number of different analytical pipelines. For example, on the top level, looking at both germline and somatic alterations from simple point mutations all the way through CNV, structural variants, and loss of heterozygosity. On the bottom panel, the tumor-only RNA-seq also aligns through Churchill. We evaluate these data based on gene expression using salmon transformation, and then evaluate the resulting RNA-seq in a variety of ways. That includes things like fusion detection, which I'll talk about briefly, variant calling, but in the context of the tumor microenvironment, we evaluate the immune cell infiltration using estimate, TME evaluation using CyberSort deconvolution, or for degraded samples, the NanoString platform. We also perform single sample gene set enrichment analysis, and in some cases, we may also identify neoantigens in the context of the tumor microenvironment changes. And I'll show examples of all of these from some of our real patient studies. Finally, just to illustrate what we've been up to, over 50% of the samples that we've sequenced and studied in the way that I just described so far are from the CNS space, and this colorful diagram just shows you the differences and proportions of those WHO classifications that I mentioned at the beginning, with the preponderance being astrocytic and oligodendroglial tumors, embryonal tumors, and so on, as you can read from left to right on this slide. So we are seeing a variety of different types of common and rare brain cancers. And today what I'll focus on is just some of the studies that we've done on these small number of longitudinal cases, as I mentioned earlier, looking at different CNS histologies and the differences in the tumor microenvironment with some of the tools that I just described, and we'll go into here in a little bit more detail. One of the challenges in pediatric cancer is that some of these cancers are quite rare, and so we don't really have great diagnostic mechanisms, and in some cases, the diagnosis is uncertain through the pathologist's microscope. So one resource that we found to be quite valuable is the Treehouse Childhood Cancer Initiative. This is housed at UC Santa Cruz in David Hausler's lab and is overseen by Dr. Elaine Mardis. And you can see the URL at the top of this display for the Treehouse Initiative. This is a unique resource in that it provides a large number of de-identified RNA profiles for vast number of different tumor types, including many CNS cancers, as well as tools that allow one without any sophisticated bioinformatics knowledge to simply import a list of the top 500 expressed genes into the UCSC tumor map and compare your sample that you're under study to a variety of other tumors that are housed in this database. And I'll show an example of how this is done. Another approach that we're using with RNA-seq, again, is gene expression outlier analysis. There are a variety of approaches that one can use here, including the RNA-seq data analysis using salmon. We then obtain the TPM values, and we compared our data to the Treehouse database, again, using this decision tree approach that's shown on the left-hand side of the slide. So here we can either compare our data to the IgM patient cohort, which has been growing over time, over 250 patients at this point in time, or in the past, Well, before we had so many patients in our database, we could compare directly to the treehouse cohort that I just described and get lots of information about gene expression and principal components-based grouping with different histologies. The other aspect that comes out of this type of outlier analysis is shown in the orange boxes along the bottom left is evaluation of gene expression outliers that may be indicative of druggability. So genes that are being overexpressed may also be overexpressed as protein drivers of the cancer. And in some cases, there may be appropriate FDA-approved drugs for these patients to have considered in their treatment. More recently, we've changed from this bulk approach to evaluating outlier expression to a more refined one that acknowledges the different types of histologies that we've been studying. So this is just one example of comparing a given patient, CH198, shown in the red diamond, which is from a Ewing sarcoma patient, to essentially other Ewing sarcomas in the box and whiskers plot, but also across different cohorts within the treehouse shown here. Or as I mentioned, we can also take a look at specific DEC gene expression changes for this patient. I'm looking and focusing here on JAK1 expression. We can see that compared to other non-CNS tumors, this patient has a very high expression of JAK1, and just by comparison to other tumor types, this is the level of JAK1 alteration expression in heme malignancies shown in the blue dots. So again, indicative of outlier expression in a druggable target for this patient with Ewing sarcoma. Fusions, again, are very notoriously common drivers in pediatric cancers, and unbiased approaches like the one I'm showing here can actually also help us to identify novel fusion drivers and novel fusion partners. And this is not yet a clinical sequencing pipeline in terms of interpreting the RNA sequencing data for fusions. As I mentioned, we have the Archer, which is more of a known fusion partner-based approach. But here we're just, again, taking the same data set from RNA sequencing that I talked about a minute ago for differential expression, and instead running it through a multi-component, multi-algorithmic pipeline to evaluate for different fusions. The sheer number of algorithms here is indicative of the fact that fusion detection is difficult, and certain algorithms perform better than others. As a result, we perform lots of different approaches and then run an ensemble overlap script to determine which fusions are predominantly called by more than 3 of these algorithms that are shown here. This actually has a very high yield in terms of true positives when we then take the proposed fusions through a secondary clinical confirmation-based approach. So we know that this is computationally expensive, but definitely worth it in terms of coming up with very high-value, true positive results. More recently, rather than detecting just fusions, it's also come to our attention that the detection of internal tandem duplications in cancer genes is very poor in general. And certainly in our pipeline, we felt that we were probably missing a lot of these internal tandem duplications. And so more recently, we've added the Cicero algorithm, which comes from St. Jude, for the express purpose of ITD identification. Now, Cicero also will detect fusions, but it is equally well-powered to detect these ITDs. And this is now brought into the mix of what we're trying to do to work towards a validated, clinically oriented pipeline for RNA sequencing evaluation of fusions as well as internal tandem duplication. Here's just an example of a recent result of an ITD detection. Using Cicero in this patient, the internal tandem duplication is in this known cancer gene, FGFR1, for which there are a variety of molecular targeted drugs. And you can see the diagram of the insertion on the left-hand side of this slide, and on the right-hand side, the corresponding differential sequencing expression analysis of the FGFR variant that's been identified. So we know that this can add value to the RNA-seq data that we're generating from these patients. And of course, for those of you who are interested in neoantigens, it's also the case that many of these novel sequences, essentially for fusions as well as for ITDs, may present indeed very powerful neoantigens in the context of that type of analysis and use for potentially neoantigen vaccines. So a more well-accepted and widely used approach to studying the tumor immune microenvironment from RNA sequencing data is shown here, the CyberSort, or more recently CyberSort X algorithm. This is really a pattern matching approach where the normal gene expression profiles of immune cell subsets are known. The RNA-seq data is matched to that, and a significance threshold is evaluated through permutation analysis to return a result on the relative global infiltration of different immune cell classes. And this is something we're using quite readily, and I'll show you some examples. Sometimes, however, RNA comes to us in a fairly degraded state, often from improper preservation, time in formalin, or other aspects, and we don't have a high-quality RNA to study through conventional RNA sequencing. For the tumor microenvironment profiling, however, we can use this nanostring immunoprofiler assay shown here. I think people are probably familiar with nanostring, but it's essentially using a fluorescent molecular barcode to identify different genes through specific probe hybridization, alignment onto the silicon surface, and then a readout of the different the different RNA expression profiles, rather, that correspond to different immune cell types. And so I'm working through this analysis approach that's provided by nanostring. We can identify 24 different immune cell types and populations, as shown on the right-hand side, that correspond to a variety of different actors in adaptive, innate, humoral immunity, and so on. So this can actually be quite a valuable tool, as I mentioned, when the RNA quality is not there. Finally, I was really pleased when I was at WashU to be a part of the development of PVAC in 2016, and then more recently this year of PVAC Tools, which is really an enhanced approach to neoantigen prediction, where all of the different variants that one can discover through the types of pipelines that I've been describing, including fusions, ITDs, point mutations, et cetera, can be run through these variety of different tools and to evaluate the potential neoantigen presentation by different types of variants. Just a plug for the pvactools.org approach to neoantigen prediction. And again, I'll show you some results from this in a moment. So if we look broadly at our cohort of cancer samples through all of the different tumor types that have been studied in our protocol to date, this just gives you a snapshot of nanostring pan-cancer approaches, where on the left-hand side, we're separating out some control brain normal samples, just a couple shown there, compared to CNS cancers, heme malignancies, and non-CNS cancers as just broad classes. And on the y-axis are just the total number of tumor-infiltrating lymphocytes, or TILs, where you can see, especially for CNS cancers, because we have so many, that there's a significant spread, if you will, in the tumor-infiltrating lymphocytes that are identified in our CNS cancer population. If we do that same type of analysis using CyberSort deconvolution, now just focusing on CNS tumors as a class, you can see here across the broad swath of different tumors that have been approached by CyberSort RNA-seq deconvolution that we get a variety of different results for all of these different T-cell, macrophage, dendritic cell classes that are shown in the colored boxes on the right-hand side of the slide. So this just gives us a general feeling that there's a lot of differences and similarities in some cases just across the CNS cancer cohort that we've been able to put together so far. So what have we learned from these longitudinal samples? So studying the pediatric to the recurrent cancer across the tumor immune microenvironment changes that go along with a recurrent brain cancer. Well, the answer is that the changes actually differ somewhat depending upon the type of brain cancers. And I'll illustrate that here with a few examples, extremes, if you will, just to really drive the point home. So one of the most common types of brain cancers are low-grade cancers called pilocytic astrocytomas. These are typically fairly indolent cancers that grow slowly for long periods of time, often get detected by the presentation of specific symptoms in patients, identified by imaging, and removed, if possible, in their entirety by surgery. And this is just one example of a patient, a 3-year-old male, where the primary pilocytic astrocytoma was diagnosed in 2017, and then there was a recurrent tumor detected in 2020 through routine imaging. Using our fusion analysis, we identified a common fusion driver in pilocytics, which is the KIA-BRAF fusion. This is well known and this fell into a classification of KIA-BRAF fusions that had been identified before. If we look at a gross scale score of immune infiltration using the estimate algorithm, which we often do, again based on RNA-seq, but not with the level of granularity of CyberSort, which we'll talk about in a minute, we can see that comparing the immune score from the primary to the recurrent, there's really not much of a change in terms of the numbers there. So not really a significant difference in the amount of immune infiltration. And the Ki-67 is just a marker of aggressiveness or cell division, and this shows that this is an indolent tumor because the Ki-67 is below 5% for both the primary and recurrent cancer. So what you might expect is that there are not significant differences between the primary and recurrent TME, and that's in fact the case shown here. The primary result is from CyberSort is shown in the top panel, the recurrent in the bottom panel. And even though one is larger than the other, when you really look at the specific absolute values for each of these different classes of immune cells, there aren't significant differences that are identified from the recurrent primary to the recurrent tumor. So let's turn our attention to a different case, which is also has a different result. And this was a bit of an indeterminate diagnosis, which I mentioned earlier. This is a patient with a CNS cancer detected in 2015 that was taken by gross total resection, then was detected as a recurrent tumor 2 years later and also removed by gross total resection. And we studied both of these tumors. Interestingly, under the pathologist microscope, and even by the principal components analysis shown here, you can see that these two tumors, although they're from the same individual, don't really seem to have exactly the same overlap in terms of the gene expression by principal components in the diagram. And indeed, from the pathology diagnosis, they did not obtain the same diagnostic result from pathology. And in fact, in the recurrent setting, one of the samples was even sent out to St. Jude for a second call, and that came back as a different diagnosis. So the EPIC array was also used to see if we could categorize these primary and recurrent tumors into a category according to the CNS classifier. That also was not successful. So this is a bit of an outlier tumor set, if you will, in terms of those solid WHO-based classifications that we talked about earlier. And then finally, again, from the fusion analysis, this was diagnosed with an EP300 B-core fusion that was confirmed clinically, and it was present in both of the samples. And at the time, back in 2017, was not understood as a known driver. So we made notes of it. We studied the RNA expression and other aspects, as I'll tell you about in a minute. And we sort of set this aside because it was not clinically actionable at the time. One of the things that, again, we can do to compare the primary to the recurrent cancer, shown on the left part of the slide, is to just look at the mutations, at what's shared and what's new in the recurrent compared to the primary. And here what we can see is that for the primary tumor on the x-axis, there are a large number of unique mutations, but a much larger number on the y-axis that are unique to the recurrent tumor. Only 2 that are shared. Those are the ones sort of here in the center, JADE2 and PM20D2. So much larger number in the recurrent tumor. And then when we take those mutations and we evaluate through PVAC-seq, you can see that the primary tumor really only results out one reasonably strong predicted neoantigen here, whereas the recurrent with a higher number of mutations also reflects a higher number of predicted neoantigens with the binding affinity cutoff that's shown here. And again, these are just predicted, but based on the combination of DNA and RNA evidence, as well as the evaluation through IEDB. What do we learn from the TME-based evaluation? Well, again, here we have estimate scores shown for the primary on the left, the recurrent on the right, and you can see that the immune score goes from negative in the primary to highly positive in the recurrent cancer. Similarly, in CyberSort evaluation down below, top level is primary, bottom level is recurrent, and we can see now significant changes here in two ways. First of all, T-cell CD4 naive cells have decreased significantly, and importantly, M2 macrophages have increased. M1 and M0 macrophages, as well as monocytes, are now present in the tumor, whereas there's really no statistically significant evidence for them in the primary cancer for this patient. So a good immune infiltration of macrophages of different types and monocytes. One clear question is, how does this relate to anything that we know about pathology-based staining? And so I went back to Dan Bouet, our neuropathologist, and asked him to perform some stains with immunohistochemistry just to substantiate what we had found or refute it accordingly. And what you can see here are a variety of stains for CD45, so general immune cells, CD14 monocytes, a macrophage, and then specific for M2 macrophages, CD163 cell surface marker. On the lower panel is the primary tumor, and then on the top panel is the recurrent tumor. So for all of these different classes, of course, we can see that there's a significant increase in the brown staining, which is the immunohistochemistry stain. Finally, I want to just talk about a patient here with a very rare tumor type that we don't see frequently, even in adults, but came to our attention recently. So you can see from the clinical imaging history of this patient, as you read from left to right, that this patient had a presentation and diagnosis of a brain cancer in 2019. This was removed by gross total resection. And then a follow-up imaging just a month later showed that the tumor had returned. The patient then went through a therapy intermediate of veliparib, followed by temozolomide and radiation. And the follow-up in January of this year showed that the tumor had continued to grow. So the patient was enrolled onto the cancer protocol. Our diagnosis was actually quite surprising in that this patient has Lynch syndrome, a gene called PMS2 that is mutated in the germline and also mutated in the somatic setting. And these alterations in the Lynch syndrome DNA repair aspect lead to an ultramutated glioblastoma with over 14,000 mutations in the coding regions of the exome that were identified. Many of them in cancer genes, interestingly, I suppose, but really it becomes sort of the detritus of DNA repair defects that wins the day in terms of the mutational profile for patients like this one. And this result of PMS2 germline, of course, also incurs genetic testing for other family members. What did we find when we looked at this patient from the immunogenomics and pathology standpoint? Again, we can see the different immunostains here on the left-hand side of the slide. At the bottom, the primary tumor stained with CD163, again, the M2 macrophage stain. These upper three sections are all stained also with CD163, and they just represent different portions of that recurrent tumor that was taken in October of last year. And what we can see here is, again, a very similar story, especially in this sort of yellowish shaded box for the M2 macrophage population, differing, of course, based on different tumor cellularities, but most expansile here in this portion with 60% tumor cellularity when compared to the highly non-involved primary tumor with about 99% tumor cellularity, of course, indicating that there were very few infiltrating immune cells under the pathologist microscope on H&E. So this is again reflective probably of the significant mutational and neoantigen burden in this setting for this patient with this ultra mutation, and also the fact that in the recurrent setting, the return of that tumor brings in immune cells from the peripheral circulation, probably setting up a immune suppressive environment, which is a known attribute of macrophages in the M2 setting. So we can see just again some more analysis here for the same patient that I've been showing you. I'm comparing the primary in the pink dots to the other 3 recurrent samples that were studied in the aqua, green, and gold dots across different immune cell classes. So generally comparing CD45, a general immune cell marker, highly elevated here. Macrophages we already talked about. Neutrophils are fairly similar. Th1 cells are really T helper cells, the only ones where the situation is reversed, so higher in the primary tumor than in the recurrent, and so on across the different cell types that are being evaluated here by the nanostring predictor. And if we look now at aspects of immune checkpoint expression, you may have noticed on that first slide for this patient that he is now being treated with nivolumab, an anti-PD-1 inhibitor. You can see here that the PD-1 counts are elevated in the recurrent cancer over the primary, although you could argue that they're roughly the same. PD-L1 also elevated, CTLA-4 significantly elevated, and ICOS remains basically the same. So again, sort of evidence that there is immune suppression in place. And if we can remove that immune suppression using an anti-PD-1 antibody, this patient may be able to battle recurrent recurrences of the cancer in the CNS space. And at this point in time, based on recent imaging, this patient continues to do well in the setting of treatment with nivolumab. So I just have a couple of minutes to finish here with some new tools for immune microenvironment studies, which is digital spatial profiling. So everything I've shown you has basically been from bulk tumor sequencing, where we mush up the tumor, extract the DNA and RNA, and lose a lot of the spatial information. And this becomes somewhat important in the setting of treatment with different immunotherapies. So more recently, we've been utilizing the NanoString Geomics digital spatial profiler. This just shows you sort of a 1, 2, 3 through 7 stepwise approach where we are essentially taking formalin-fixed slides of different patient tumors using a detection system using up to 80 different antibodies with corresponding fluorescent probes. We image these onto the Geomics platform, identify regions of interest, and then use UV light to cleave the oligos off of the antibodies that are staining those regions of interest. These are removed then from the surface, placed into a 96-well plate, hybridized, and then placed onto the end counter for sequencing and counting. And then we marry those ROIs back up with the information from the different antibodies that were bound there. This is just sort of showing the different 43 protein immuno-oncology panel, along with the control probes that are utilized. Here, we use the solid tumor TME morphology kit to identify a pancytokeratin stain, a CD45 immune cell stain, and then a nuclear stain. And then using these different probe sets, we can identify different types of infiltrating tumor cells across the tumor immune microenvironment. We've recently just started using this instrument, and so I just have a few results to show you, but we've studied one patient in the recurrent setting, initial medulloblastoma that recurred as a radiation-induced glioma, so a completely different tumor, but in the same space in the CNS. And that this is the patient that I talked to you about a minute ago, the neural epithelial high-grade glioma in the setting of PMS2 loss. So let's take a little bit of a look at these patients. Here's just a comparison of this primary medulloblastoma mutations compared to the recurrent glioma, and you can see that there's no sharing of variants between these two genes, leading us to hypothesize that this is a secondary cancer resulting from radiation treatment in this patient. When we look at these two tumors in comparison using CyberSort, you can see that in the glioma, which is the secondary cancer, a different color setting here, but again, a significant increase in M2 macrophages in this funny purple color shown here that are essentially only in a very, very small proportion, if I'm even getting the color right, and it's hard to tell in the medullo presentation from 2012. So again, a high-grade aggressive tumor that recurs, and it recruits in M2 macrophages. This is our patient with the high-grade hypermutated glioma, just to show you this progression from slide staining, identification of the different regions of interest, which are these circular areas, and then one of these shown in a high magnification to show you the staining in that region. And then the analysis that comes out of this is really shown here. So again, a region with some CD45 staining, some pancytokeratin, here with a large amount of CD45 staining. And what we can see here is that when we break this out into different categories, in particular for this region of interest, we're seeing a very large amount of CD163 staining, again, the M2 macrophage signature. And this just shows the total analysis for these different tumors. 153, the hypermutator shown here. A primary tumor for 42, the medullo, and the recurrent glioma with an uptick in granzyme B activity reflective of the higher immune activity in this tumor. And this is just showing you that basically the comparison of CD163 counts from regions of interest taken from the primary medullo and now from the recurrent high-grade glioma in this patient with the recurrent radiation-induced cancers. So to finish, I would like to just conclude that these recurrent high-grade CNS cancers seem to have multiple lines of evidence, which I've tried to show you, for macrophages and monocytes in the TME that are recruited from the periphery. They do seem to be present, but only minimally in the primary cancer tumor microenvironment. So this is something that goes along with recurrence. And these are likely providing some sort of an immune suppressive context based on the evaluation of checkpoint blockade expression. The presence of neoantigen encoding alterations may also present an avenue to engage the immune system. And we have to, in the setting of immunotherapy, keep in mind that our small patients actually have very grave differences in their immune systems that are age-based. And so taking this into account, we've started thinking about more novel approaches to immune therapy than checkpoint blockade, which can be efficacious in some cases, but is probably too aggressive for these young immune systems. And to this end, and with some of the data to support it that I showed you today, we have a recently funded U54 from the NCI that will start exploring new immunotherapy approaches for pediatric cancers. These include the use of oncolytic viruses, as well as engineered natural killer cells. And hopefully I'll have a lot more to talk about in that regard in the near future. So I'll just finish by thanking you for your attention, thanking a very large team of investigators at the IGM that are listed here, my co-executive director, Rick Wilson, and certainly acknowledging our computational analysts, clinical directors, and importantly, our oncology collaborators who are listed out here, who are wonderful and enthusiastic about the things that we're doing. And finally, a special thanks to our patients and families for their courage in consenting onto this protocol and helping us to develop new hypotheses about ways to defeat brain cancers. Thanks so much.
All right. Thank you so much, Elaine, for the wonderful, wonderful talk. This is really, really impressive. So I see that there are questions coming in, so I'll just read them to you. First question from Lars Eriksson from Catskills Research Company. The question is immunotherapy applicable to benign tumors such as optic nerve glioma? If not, what will be your approach for such tumors, such benign tumors?
Yeah, so these actually, we were just looking at one last week in Neuro-Oncology Tumor Board. These actually do present a significant challenge I don't know how much is known about the genomics of these tumors, and I think that's probably where I would start, obviously. But if it were evidence were accumulating of the type that I showed today in terms of recruitment of different immune classes, one might be able to consider specific immunotherapy intermediates. The other possibility would be something along the lines of a personalized vaccine, or indeed, if there were shared antigens, some sort of a shared antigen-based vaccine. But I think they haven't been studied so much, largely because these are sometimes in the space where they can't be removed or even biopsied safely, which makes it a big challenge to study the genomics, to be sure.
Okay, great. So I have a quick question. So Elaine, you show that there's one case that you showed, that this JL36 case, by comparing primary and recurrent tumors, and you show that you can quantify immune infiltration from the omic data, and then you compare with the pathology images. But in this line of work, is there some potential directions that you can more coherently integrate these two type of modalities, right? Because because sometimes the complementarity or the signals from each one of these modalities is subtle by eye, but perhaps if you put them together, it's going to be more revealing.
No, it's a great point, Gian, and I'm glad that you asked that, because that's absolutely what we're after here, if it wasn't obvious. And I have to say that one of the challenges, of course, in the conventional pathology setting really is the amount of tissue that's available for doing staining and the ability to do repeat staining. And this is why I really am excited about these digital spatial approaches. Not only do they tell us about whether the immune cells are sitting at the periphery of the tumor or whether they're actually infiltrating in, which can be really important information, and whether that changes with the therapeutic intermediate, which is what we're after in this U54 grant. But it can also give us the ability to really do a couple of things. One is to identify and label antibodies that correspond to commonly overexpressed RNAs. And one of the challenges that we have is, if it's overexpressed as an RNA, is it also overexpressed as a protein, and therefore is it a druggable target? So I think the genomics will, absent spatial aspects, which of course are potentially important, it will also tell us if there's a tumor cell, does it have an antibody labeling it or more that are indicative of that particular target being overexpressed? And can we ask that across a number of different potential targets? The challenge in pediatrics is really often that we don't have a lot to go on in terms of what's driving the cancer. This is one of the reasons we study RNA and look for fusions, et cetera. But the challenge is that when you find something that looks like it's real at the level of RNA, you often have to reflex to a protein-based endpoint to really substantiate that that is a therapeutic target that's overexpressed and therefore valuable for consideration. So I kind of see the fluorescent approaches as giving us, if you will, the new pathology. And I don't think that's a threat to pathologists, because I have a lot of friends in pathology who are also really fundamentally excited about these technologies and what they're going to do in terms of really getting the value added yield out of, just a single tissue slice. So that's kind of the way that we're thinking about the integration and the cross comparability of these different data types right now.
Great. So Elaine, really the last question, and I, since you're here, I would like to ask you a question, maybe a good segue to the next activity item after your keynote speech, is that I know that a lot of the students in the audience, they are from a computational background. They're computationally savvy, and I know many of them are interested in cancer genomics and cancer biology, but in the meantime, they also find it very daunting, a little intimidating. And I know that you have also transitioned some very successfully. I know Ken Chen very well. That he had a hardcore signal processing background, but you transitioned him into become a leader in cancer genomics. So maybe you can give especially some of the young students in the audience, some tips or some suggestions if they're interested in getting into cancer systems biology, what type of preparations or what kind of general suggestions you have?
I think that's it's a really nice thought, Jan, and hopefully what I say will be helpful and impactful. I basically, my whole career has been in team science, and I really see, especially now in this clinical translational setting that I talked about today, the value of team science. I think the challenge in computational genomics, and especially in cancer computational genomics, is really getting involved with the right team. And so that's what I've tried to do for people like Ken Chen and others who I've mentored over the years in the context of people who really understand hardcore math and hardcore computational programming and algorithm development, machine learning, et cetera, and trying to get them excited about the problems that we have in terms of identifying fusions, for example, or figuring out how to classify a patient as MSI unstable versus stable, something we recently did with a machine learning based approach. My best advice is to look around, look in the literature, find teams that are doing exciting, impactful work that is in the computational or systems biology realm, and try to connect in with them. I think the challenge always in computational studies is that you want to be valued for the contributions that you bring. So make sure that that's the case when you join these groups, and don't just feel like you're the go-to red button bioinformatics guy. Here's some data, give us a result kind of a thing. So it has to be a healthy team environment, but there's so much interest nowadays, it's almost hard to not be valued in that setting. So I would just say find hard problems, people that are trying to address them, and have the data and especially the link to patients to really make it meaningful. And that will put a context on the capabilities that you bring to the table.
Great. With that, so on that note, I think thank you again, Elaine, for joining us and giving us a great, wonderful keynote speech. And thank you all for joining this session. I think we're going to transition to the next item, right?
Okay.
Network.
Great. Thanks again for the invite. Take care.